All right, today we're going to unravel the incredible story of a protein that's right at the very center of Parkinson's disease. Its name is alpha-synuclein, and it leads this kind of double life inside our brains. It's a story that's part hero and part villain. So to really get this, just think about this question for a second. What if a hero, a really essential worker inside your brain, just turned into a villain? Because that right there, in a nutshell, is the story of alpha-synuclein. Okay, so first things first, let's meet the hero. In a perfectly healthy brain, alpha-synuclein is absolutely one of the good guys. It's a master coordinator, basically acting as the brain's traffic controller. So what is it exactly? Well, alpha-synuclein is a small but super common protein, and it's just packed into the very tips of our neurons. And what's so wild about it is its shape, or actually its lack of one. Scientists call it intrinsically disordered, which is a fancy way of saying it's kind of like a flexible ribbon, just floating around until, bam, it bumps into a cell membrane. And then instantly, it just snaps into a specific working shape. Its main gig is to manage these tiny little packages called synaptic vesicles. These are the things that carry crucial chemical messages like dopamine from one neuron to the next. Alpha-synuclein makes sure those messages get delivered on time, nice and smooth. I mean, it is absolutely essential for every single thought you have, every move you make. But then, something goes really, really wrong. A single chemical event happens that acts like a switch, and it flips our hero protein onto this dark, destructive path. And that event? It's called phosphorylation. Now, this is a totally normal process. Just think of it like your cells adding a tiny chemical tag, a phosphate group, to a protein. They do it all the time to turn proteins on or off. But in the case of alpha-synuclein, this switch gets flipped and then it gets stuck, permanently stuck in the wrong position. And look at this, because this slide really tells the whole story in just 2 bars. In a healthy brain, only about 4% of all the alpha-synuclein has this chemical tag. But look at the brains of people with Parkinson's. Specifically, inside those toxic clumps called Lewy bodies, it's over 90%. I mean, this isn't some small change, it's a complete chemical takeover. So, the really important question is, how does one tiny little tag cause so much chaos? Well, when that phosphate group latches onto one specific spot, it's called serine 129. It completely changes the protein's electrical charge. That forces it to fold into the wrong shape. So that flexible ribbon, it becomes stiff. And even worse, it becomes incredibly sticky. And what happens next is what's truly terrifying. That one sticky protein, it doesn't just sit there. No, it kicks off this devastating chain reaction, a cascade of damage that just starts to spread all through the brain. It all begins with just one, that first sticky, misfolded protein. It starts grabbing onto healthy ones, corrupting them, making them misfold too. Then they start to clump up into these small, super toxic clusters called oligomers. These oligomers then start linking together, forming these long, ropey chains called fibrils. And eventually, all those fibrils get tangled up with other junk in the cell, creating these huge roadblocks that we know as Lewy's bodies. And these clumps are anything but harmless. They basically wage a war on the neuron from all sides. They go after the mitochondria, the cell's power plants, causing this kind of cellular rust. They totally clog up the cell's trash disposal systems, and they get in the way of the original protein's job, which makes dopamine leak out and become toxic inside the cell. It's just a total disaster. This chart just perfectly illustrates the 2 completely different fates of this one protein, right? On one side, you've got the hero. It's soluble, it's dynamic, it's doing its job. On the other side, you've got the villain, an insoluble, phosphorylated clog that just gums up the works and eventually leads to the death of that neuron. And maybe the scariest part of all is that this problem spreads. That toxic, misfolded alpha-synuclein can actually get out of a dying neuron and be taken up by a healthy neighbor. Once it's inside, it acts like a seed, kickstarting that entire clumping process all over again. And that is how the disease just marches relentlessly through the brain. But here's the thing. This incredibly detailed understanding, it's also the source of our greatest hope. Because by knowing exactly how our hero becomes a villain, step by step, scientists now have a precise blueprint for how to start fighting back. And this is where the story starts to turn again. Because we can identify the villain, we can now go after it directly. We can create tests to hunt for this very specific phosphorylated form, which could lead to much, much earlier diagnosis. We can design antibodies that work like little heat-seeking missiles to find and clear out those toxic clumps. And we can even create drugs to block the enzyme that adds that first phosphate tag, which could stop the villain before it's even created.
As researchers have pointed out, while this disease is unbelievably complex, that very complexity gives us multiple ways to attack it. Every single step in this destructive process is now a potential target for a new kind of therapy.
So this leaves us with one final, really powerful question. Now that we understand the exact switch that turns the hero into a villain, could we one day figure out how to intervene and stop this protein from ever breaking bad in the first place? That is the ultimate goal. And for the first time in this long, long fight, it actually seems like it might be within our reach.